In this video, we will discuss about CAR-T with fusion soluble proteins expression to counter TME immunosuppressions. So their main objective is focused more on the solid tumors, where uh, what they have seen or what so far has been understood about CAR-T is that they are less efficient on the solid tumors because of the immunosuppressions effect of the TME. So what they did is they fused 2 to 3 different things, and their image is shown in the panel A, where what they did is they have the regular CAR that has the antigen towards the tumors. Along with that, they have these bifunctional fusion proteins. So this bifunctional fusion proteins, this time in the previous paper, what they did is they kept it on the membrane, but in this time what they did is they make it soluble in the extracellular matrix. That is, they excrete this bifunctional fusion proteins into the extracellular matrix. And what this bifunctional fusion proteins contains, it contains this Fc region and also the antigen that can target this PD-L1 ligand that are present on the immune cells, so that the immune checkpoint, it can work as an immune checkpoint inhibition therapy, and thereby prevent the deactivations of the immune cells of the host. Now on the other case, it also has the different cytokines, that is interleukin 12, interleukin 15, and this TGF-beta trap. So this is what their whole design of the CAR construct with bifunctional fusion protein is. So first they check the expression levels of these different combinations of the CAR, and in the panel B they have shown the expression level of the different combination, that is with CAR alone, with interleukin 12 and this PD-L1 and this mutated PD-L1, interleukin 12 and interleukin 15, and different combinations, and they are able to see almost similar amount of this transduction efficiencies. And they also check the efficiencies of, not just the efficiencies of this CAR construct. Here they check the efficiencies through these CD19 expressions, and they also check the efficiencies of these fusion protein expressions through this truncated Fc that is expressed in this PD-L1, and they are able to see the Fc expressions are also similar. Once confirming that they are able to get a similar amount of transduction efficiencies of the CAR construct and also of these fusion proteins, then they did some in vitro studies. First what they did is they induce PD-L1 on the PD-L1 on this CAR T cells and tumor cells co-culture. First they induced this PD-L1 on the CAR T cells through these CAR T cells and tumor cells co-cultures, and after that they took the supernatant of the engineered T cells that contains this PD-L1 ligand, and then checked the PD-L1 expressions. So since the supernatant contains this PD-L1 ligand, so in the case where supernatant are added, they should able to see a less amount of PD-L1 ligand, as they measure their signals using the flow cytometer. And what they are able to see is that in the case where they use PD-L1 in the wild type form, the signals of the flow cytometer goes down of this CAR T cells, because this PD-L1 that are present in the supernatant will compete for the PD-L1 antibodies that is used for fluorescent measurement using flow cytometer. So that can be seen here. In all the case where they use wild type PD-L1, they are able to see a decrease in the signals of the PD-L1 binding to the CAR T cells. Next, what it is in the panel F, they check they also check whether these TGF-beta 1 receptors or interleukin 15 or interleukin 12 are expressed or not, and the TGF-beta 1 is bind or not. in these conditions, and they are able to see that all the three things, that is TGF beta 2 is seems to be bound, and also this interleukin 15 and interleukin 12 expressions are also there, indicating that the system is working as per the expectations. Then they check some in vitro studies where they co-culture these effector or target cells in 1:2, and they re-challenge these CAR T cells by re-additions of the tumor cells at 4 different intervals. And in all the cases, they are able to see the tumor death is highest in the case where they use PD-L1 ligand. There is anti-PD-L1 and interleukin 12 that can be seen in the panel H, and in the panel I, they check the expression levels of this CD3 count, and they can see the CD3 count expression levels are also high in the case where they use interleukin 15 or interleukin 15, whether it's PD-L1 mutated or PD-L1 non-mutated. Then they check the CD19 expansion, or CAR T expansions through CD19 signals, and they are able to see the CD19 signals are high when interleukin 12 is used. And when they also check this co-stimulatory formations or translations, and in the case in which they see the maximum co-stimulated translations through this 4-1BB is in the case where interleukin 15 is used or interleukin 12 is used with this PD-L1 ligand is in wild type form. And thereafter they also check the signals for this TIM3, and which is kind of signals for the high activity of the CAR T cells, and they are able to see it is high in the case where interleukin 12 is used or interleukin 15 is used with either PD-L1 is mutated or PD-L1 is present in the wild type form. Then they check the IFN gamma expression levels. IFN gamma expression levels are also high in the case of where they use interleukin 12 along with PD-L1 ligand, or where they use interleukin 12 with mutated PD-L1 ligand. So all this comes to a point that from the in vitro studies, they are able to see some positive results, but there is no uniformity or that they are able to find out. So in order to check whether this uniformity means whether the PD-L1 ligand is beneficial or this PD-L1 anti-PD-L1 fusion proteins is not beneficial. In order to find out that thing, they need to do in vivo studies where they will get a better idea. So from these in vitro studies, they can see that the effect of the cytokines, but there is no uniform results about the anti-PD-1 ligand that are also secreted by their CAR T cells, engineered CAR T cells. So in order to find out whether they have any effect on this CAR T increased efficiencies or not, they have to do the in vivo studies. So far in the in vitro studies, or in this figure, they presented these results. If you need to understand more, I hope you understood to some extent what are the experiments that they have conducted to validate their CAR T cells, engineered CAR T cells, along with these bifunctional fusion proteins. If you need to understand more about their experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.